Now we're going to address disease resistance of the lymphatic system. Now, the immune system does protect the body from foreign substances and harmful chemicals that can damage the body. We're going to be learning a lot of these terms. One of the terms, and the most common one that we're going to address, is a pathogen. A pathogen are viruses, bacteria, or other microorganisms such as protists or fungi that can cause a disease. Here's a list of terms that you will need to know. We have a pathogen, which is a disease-causing microbe or microorganism. We have infection, when the pathogen invades the body or enters the body. A disease is the illness characterized by the specific symptoms created by the pathogen or the infection caused by the pathogen. An antigen is the foreign invader that causes a response. And so this can be anything from a virus or a bacteria, or even a foreign blood type or tissue. An antibody is the protein that your body has. So this is the protein produced in response to the foreign antigen. It protects your body from the foreign invaders. A hapten is a smaller antigen. This can be like what your body is going to react to with certain allergies. So it's so small that another molecule has to attach to it, so it'll be recognized as an antigen. And then virulence. This is the power to cause a disease. Now there are non-self antigens, which are your normal antigens that we think of. This is outside the body. These are things that are going to, as we said, provoke this immune response that enters the body, such as foreign proteins, and as we talked about allergies, pollen grains, and other microorganisms. But a self-antigen is primarily going to be something that is going to trigger an immune response in your own body, maybe an organ transplant. So because human cells have their own surface proteins, you're not going to attack your own body. But if there's a transplant put in your body, an organ or a skin graft, this can trigger an immune response against part of the new body organ or tissue that was added. Now, there are two types of immune responses that we're going to focus on. There's nonspecific and then specific immune responses. Now, the main difference between the two is that nonspecific immune responses stand the same against all pathogens. So it's not specific to a specific type of virus or bacteria. It's just very general. We're going to go into more detail about this, but this can be like the skin. This can be just a physical response, or the enzymes or chemicals in your stomach or in your tears. It's going to stand against anything or as many different pathogens as possible. It's not specific only to the coronavirus. So it's very general. That's non-specific. While specific immune responses are going to protect the body against a specific type of virus or bacteria, while specific immune responses are generally going to be adaptive or acquired immunity. And we'll talk about what acquired is in just a little bit. We have active immunity. This is where you're going to have your B cells encounter certain antigens and they're going to produce antibodies. Active immunity is usually natural or artificial, and we'll talk about that in a little bit. Then we have passive. Passive immunity is when the antibodies are obtained from someone else. So your body did not produce them. This can be generated or passed on by the mother to the child. This can be artificially transferred to your body through a immune serum. So the focus here is for passive immunity, they do not have a memory. So you have acquired immunity, as we said, natural and artificial, active and passive. Active, you get the infection. This is naturally acquired. Active, you get the infection, and then your body produces the antibodies. Passive, naturally acquired. Antibodies pass from mother to child, so that those two are both naturally acquired. So active, naturally acquired, you get the disease, then you make the antibodies. Passive, you pass on the antibodies from mother to child. Then we move to artificial, and this is going to be artificial active. This one is going to be the vaccine. So you give the vaccine the weakened, broken pieces of the virus, and your body codes for it, figures out how to read it, and figures out how to destroy it. That is an active, but it's artificial. So we injected it into our body. Passive is when you inject, as we said, you're injecting the antibodies directly into your body. So the inborn or innate immunity versus the acquired immunity, as we said, innate is going to be you're born with it. It's passing through your genes. This can be a result of your genetics, what you pass on through a parent to child. This doesn't usually involve the production of antibodies or cellular immunity. So this is what you're born with. Now, acquired, we said, is what you're exposed to. This is something you're getting. So we have 4 different types. We have natural, acquired, active immunity. So natural, acquired, and then active. So now we're taking quite a few of these terms that we've just talked about. Natural, okay, versus artificial, acquired, and then active versus passive. So natural, acquired, active immunity. So you have an exposure to the antigens, you get the disease, so you won't get it again. It's a long-term immunity. Natural passive is, we said, mother to child. This could be antibodies are in the milk or going through the placenta to the baby. Artificial active is through the vaccine, the shot. You get the weakened virus. Artificial passive, we said, okay, the acquisition of the antibodies directly in the body through a serum. So we're going a little more detailed in the names here. So let's talk about the nonspecific disease resistance as we spoke a couple slides ago. This is a resistance to all diseases, and we said it's not specific to one specific type. It's more broad or general, and it doesn't have a memory to remember which virus is coming in or bacteria is coming in. It's just very general. So this is going to include fever and inflammation. And we'll talk about that on the next couple slides. So you can have a lot more information on the next couple slides about the fever and inflammation. But inflammation is a redness by vasodilation. So the veins, the arteries, basically dilate, and this is caused by histamines released by mast cells. So let's first talk about the first type of nonspecific disease resistance, which is mechanical. Mechanical is also known as physical. So these are physical barriers that protect the body. Here are some examples. The skin. The skin does have an acidic pH to prevent bacterial growth. It prevents and stops things from getting into the bloodstream or into the muscles or bone, into the nerves. So the skin is a first line of defense. Then we have the saliva, which contains enzymes. Mucus, which will trap particles because of the thickness. Think of snot. This will trap particles and then you can blow it out. We have cilia in our lungs to move particles out of our body. As you cough, it starts trying to bring up the particles up out of your lungs. We have the tears. The fluid tears is a mechanical and a physical protection. The physical is the flushing out of the pathogen or the material that should not be in the body. Maybe you get something in your eye and you're tears try to flush it out. So that is physical. And then we have waste elimination, such as going to the bathroom, pooping, excreting out feces. And then we have regurgitation, throwing up. When you get a stomach bug, sometimes you throw up to eliminate, get rid of that virus out of your stomach. Now we have chemical protection, and these are chemical agents such as the oil found on your skin, and that is sebum. It's also in your ears and on your hair, your scalp, and that is toxic to bacteria. We have the lysozymes or lysosome. Now, these are going to be enzymes that break down. This is found in the saliva and also in your tears. And this is going to lysis or break down the bacteria. And then we also have gastric juice or hydrochloric acid and pepsin, which is in your stomach, and this breaks down bacteria as well. Now, kind of jumping back to the broad spectrum of your physical responses, the inflammatory response, we said the inflammation, the vasodilation, this is when the body is injured, the body tissues are injured. As you can see in the diagram, chemicals, histamines, will be released, and this causes the blood vessels to dilate, white blood cells, monocytes, neutrophils coming to the aid here. Now, 4 signs of this is going to be swelling, pain, heat, and redness. And this usually leads to a fever is an abnormally high body temperature. And what you have is 98.6 degrees Fahrenheit or 37 Celsius is normal body temperature. So you can check if someone has a fever 3 different ways: orally, rectally, or axillarily. And that's going to be under the arm and in the armpit. The fever isn't generally a bad thing. If it's too high for too long, yes, it can cause severe damage, but high temperatures causing a fever will inhibit the release of iron and zinc from the liver and spleen. And bacteria needs iron to grow. So the focus here is by having a high temperature, you stop providing certain pathogens what they need, and they do need iron and zinc, so that is stopped. Also, the fever increases the speed of tissue repair. So those are 2 things that are beneficial for your body to have a fever. Then we're going to go to number 3, still under non-specific immunity, and this is antimicrobials. Now, an antimicrobial is basically any substance that can kill a pathogen or stop it from growing. In a general sense, antimicrobials could be a medicine, an antibiotic. It could even be an antifungal cream or a disinfectant or even a sanitizer. But when we're talking about the body and inside the body, we're going to be talking about interferons and complements. Now, the INFs, or interferons, are chemicals produced by the body cells that act on viruses. So remember, an antimicrobial is a substance like a chemical that can kill a pathogen. It can kill a virus. Now, basically, this will attach the surface receptors of the virus and prevent it from spreading any further. Now, the complements basically are a series of chemicals that will mark the virus and then send different complements to punch holes in it and basically impair it. So the C5 through C9 are the ones that mark the virus, while the C1 through C4 are the darts that punch the hole in the mark that was set by the C5 through 9 complements. You can also see the complements here as they attack and as they punch holes and they fill the virus. The virus will then expand and burst. Now, the last one that we're going to focus on for the nonspecific is phagocytosis. Remember, phagocytosis is Uh, phage or phag means to engulf or ingest. Cyte means cell. So this is where the cells are going to ingest and break down the bacteria by the use of enzymes, specifically the lysosomes that break down and digest the bacteria or the cell. A cell will surround it to release those enzymes. So the three main phagocytic cells of the body are going to be monocytes, neutrophils, and macrophages. Now, monocytes can actually divide further into the dendrite cells and the macrophages. But here's the problem. Dendritic cells are not phagocytic cells. The dendritic cells, remember, it looks like the end of an axon, it looks like a little dendrite. These are actually cells that will collect information and basically release proteins called cytokines that will notify other white blood cells to come to the site. So they will primarily let the macrophages and other cells know that there's an invader. The dendritic cells usually reside in specific tissues beneath your skin, in the lining of your nose, lungs, stomach, and intestine. But the general idea of the monocyte is that it is a white blood cell, a leukocyte, with a bean-shaped nucleus, so the darker structure inside the circle. The dark purple is the nucleus, and you're going to see some different shapes of that nucleus. And this is part of the innate. Remember, innate means inborn, or you're born with it, immune response. And basically, we're going to patrol the body for microbial cells and initiate the immune responses in times of infection. So they will respond to bacteria. So let's talk about the next two, and one of those next two will be macrophages. But let's talk about number two, the neutrophil. As you can see, the neutrophil in the picture, it has a three-lobed nucleus. Now they can actually have a 2, 3, 4, or even a 5-lobed nucleus. And this is the most abundant leukocyte, white blood cell. And this is produced in the bone marrow and then migrates to the blood. Again, this is part of the inborn immune response. And basically, most of the white blood cells that lead the immune system's response are these neutrophils or neutrocytes. Then we have the macrophages, which have little pseudopods or extensions that grab bacteria, grab little pathogens, and basically it will surround and then completely engulf and surround the bacteria or virus and ingest it and kill it, break it down through the lysosomes or the enzymes. This can not only happen to viruses, bacteria, but also fungi and protozoa. Now they are specific to each organ. So here you can see the breakdown of the different cells. We're going to start with the stem cell produced in the bone marrow, differentiate into the basophil, eosinophil, neutrophil, and monocyte. But we're talking about right now the neutrophils, the monocytes, and the macrophages as your phagocytic immune cells. So again, you can see the lobe nucleus, you can see the lysosomes inside the white blood cell, and the cell membrane. Here's the monocyte, the neutrophil, you can see the lobed parts, and the macrophage, as you can see the bacteria, and then the macrophage extending out with little cytoplasmic extensions or pseudopods. Here again, you can see the macrophage with those little pseudopods, and you can see a microscopic electron microscope picture in the top left. And that will be here in the specific immune response. So this is specific to a certain pathogen. With specific immune responses, you're going to actually have a memory, a recognition that remembers the pathogen. This could be like the flu. It may not be for life, but it may be, for about a year, maybe for a couple months. There is a certain degree of the memory. So the second time you're attacked with it, you're able to fight it off and fight it a lot faster. So some examples of this are the T cells and the B cells. Now the T cells, there are helper T cells, killer T cells, memory T cells. And then with these T cells, you're going to have, again, as we talked about, macrophages and dendritic cells are going to respond and work with the T cells and present the antigens to them so that they can be broken down. And then you have basically the B cells, which are going to differentiate into different plasma cells to produce antibodies to break apart and attach to the antigens. So let's take a look at those in more detail. Here you can see the contrast between the B cell and the T cell. The B cells mature in the bone marrow while the T cells in the thymus. The B cells are going to secrete antibodies while the T cell cytokines. And we said the cytokines are things such as the interferons and growth factors that are secreted by certain cells of the immune system, having an effect on those different cells. And they are going to provide an immune and inflammation response. Then you have the antigen receptor. We're going to talk about the Ig cells. That's usually going to be in the B cell, okay, the surface Ig, which you're not going to have on the T cell. You're going to have a specific T cell receptor. B cells are going to be found a lot in the spleen, while the T cells in the blood and lymph nodes. The B cells are going to target bacteria and viruses, T cells many times infected cells, and tumor cells. And they both have a memory, so these are specific. Here are the helper T cells, which are specific or specialized T cells that attach to the antigen, and this stimulates the B cells to produce specific antibodies. Natural killer T cells, or the killer T cells. This is a T cell, also known as cytotoxic T cells. This basically will destroy an antigen. When attached to the antigen, it's going to be activated. It's trained to recognize bacteria, viruses, virus-infected cells, cancers, and tumors, as we said on the previous page. Then we have the memory T cells. These don't respond to antigens, but they do use chemicals to mark the antigens, chemotaxis, adherence, which is a sticking or clumping together, and ingestion, or a eating or taking in, and then a digestion, which is a breakdown. And this is what an antibody looks like. An antibody has basically 4 amino acid chains. There are a heavy and light chain, and basically the heavy and light chains will form a Y-shaped molecule. You can see in the picture the heavy chains are in the purple while the light chain is on the pink. While the heavy chain is in purple or blue, and here again you can see the picture of the antibody, and you can see the heavy chain and the light chain, and then you can also see the constant versus the variable. The variable regions on both the heavy and light chain is where you're going to bond to the antigen or the pathogen. Now there are 5 antibody types. The basic Ig or immunoglobulin, this is called immunoglobulin G or immunoglobulin gamma. This is about 75% of all antibodies. This protects against bacteria and viruses by phagocytosis, and this is the only one that's going to be passed from placenta of mother to baby. Then we have immunoglobulin M, which is referring to the blood types. These are the blood type immunities. This develops in the blood plasma, and it has the anti-A and anti-B antibodies, which we'll look at in the next couple of slides. It's going to have a response to bacteria and food antigens. Immunoglobulin A is immunoglobulin alpha. This is going to be common in exocrine glands. As we've talked about exocrine glands from the skin, it comes out, such as milk, sweat, and tears. And these are going to help protect the body. Especially that's why a child, a baby, will have and receive antibodies through drinking of milk. Now, these levels of immunoglobulin alpha will decrease under stress. Now, there's immunoglobulin E, which is impacted by an allergic reaction. This is a hypersensitivity of the body to different allergens. If you may have allergies to cats, I know my daughter does, and it's really not the cat here, it's the dander, which is the skin flakes. So that would be specifically an allergy. Then you have immunoglobulin D, which is part of the surface antigen receptor on the surface of the cell. The cell surface antigen receptor on the surface of the beta cells or the B lymphocytes. So this is going to be on the surface of the B lymphocytes. Now, there is something called Rh incompatibility. We talked about the different blood types, blood type AB, and O. Now, Rh, there's Rh negative and Rh positive. Now, here's the problem with the incompatibility. If a woman, as you see in the diagram on the left side, a woman has an Rh negative. Maybe she has blood type O negative. Okay, that's the Rh. That's what we're talking about. The baby is born with O positive, as you can see in the picture. The baby has the positive. The baby received that from dad. Basically, through the placenta, some of the positive blood is going to get into the female, the mother. Well, what happens now is that the mother's body recognizes it as a foreign particle. So the antibodies trigger a response, and this is where it's actually bad, where the antibodies are now going to try and fight and break down the foreign body, and that's the baby. So the antibodies, if they get into the womb and into the baby, it could start attacking the baby and try and kill and create complications for the baby. And that's why it's important to communicate to the doctor what blood types and so forth. Now, generally, this does not impact the first baby, but it can impact the later babies because now the mother has these antibodies now. So you can see the Rh incompatibility, how basically it sends out the antibodies in the first pregnancy. So the next pregnancy, the body will specifically really go in and start attacking the next child that has this different Rh factor. Here you can see the blood types. Here you see blood type A on the red blood cell of blood type A. So if I have blood type A, I'm going to have a surface antigen, which are round. Okay, let's say they're round. Well, it will have anti-B antibodies. So if I get any blood type B in our body, basically my white blood cells, the anti-B antibodies, will try and break down that foreign blood. So if I am blood type A and blood type B gets in my body, I have antibodies to say, no, you don't belong here. We're going to destroy you. So I can only accept blood type A. But I can also accept blood type O because blood type O doesn't have those antigens that will match up with the anti-B antibodies. So I can take in blood type O or blood type A. Now let's look at blood type B. Okay, a different type of antigen. So look at the antibodies. The ends of the variable chain is going to be more circular. So if I get any blood type A in my body, the antibodies want to code and kill it and break it down. Same thing now when we go to blood type AB for those two. Now, blood type O will have the antibodies for both. So if I get blood type A in my body, it's going to try and break it down. If I get blood type B in my body, it'll try and break it down. So really the problem is, which I am actually blood type O, I am the universal donor. I can get blood type O into AB and AB, but I can't receive blood type A, I can't receive blood type B, and I can't receive blood type AB because I have the antibodies to fight against those blood types.